My topic is the PEGylation, okay? But as you know, the peptide and protein is a hot topic in the pharmaceutical industry. Everybody interested about that. But as you know, but there's no problems, okay? You know? But we must go on. Show must go on, okay? That's the why I am here, okay? And today, I'll briefly introduce my unique PEGylation platform technology that offers significantly extended biologic half-lives of peptide and small protein drugs while maintaining bioactive, okay, in vivo. A limitation, there's many limitations. It typically suffer from inherent short biologic half-lives and also low viabilities, which are common. And this is mainly due to enzyme degradations and low stabilities in liver and blood and first renal clearance. Among them, PEGylation is a gold standard method, I think so, and FDA-approved half-life extension technology. PEG protects biomolecule from degradations and increase stabilities of circulation times. Overall, it can help reduce dosing frequency, is very important, increasing patient compliance also and therapeutic efficacies. There are more than 10 FDA-approved compounds in the market. PEGylation is just a simple, okay, just attachment of PEG to the molecule, you know, okay. But PEGylation is everybody know about the PEGs. But PEG has so many different molecular numbers and also various structures PEGs on the market. For example, linear PEG is common, but nowadays dimetic and trimetic PEGs are increasing the interest for the PEGylation peptide and protein drugs. The major consideration of PEGylation. PEGylation looks simple, but it's very complicated, okay? You know, how many molecular conjugate to one peptide or protein drugs, and how to choose the size and structure of PEGs are a basic problem of novel PEGylation. But most important point is how to conjugate the PEG at the right site. This means that the site-specific PEGylation. Site-specific technique is the most important consideration in PEGylation to avoid active and receptor binding site of peptide and protein drugs. Site-specific is the most process to get homogeneous composition with highest therapeutic efficacy in the body. But no matter what PEGylation method are applied, the molecular weight of PEGs significantly dominate the PK and PD of my molecule in the body. High molecular weight PEGs like 20 to 40 kilodaltons increases, of course, PK, however, simultaneously decreased bioactivities, particularly when conjugate to the relatively small the sized molecules like peptide drugs. Therefore, it is critical to balance the PEG size with bioactivity when designing the PEGylated compound. Generally, the three representative PEGylation platforms are available. First one is the old one, is a random PEGylation. The second one is site-specific, in common nowadays. But the third is the product concept, but not yet, okay? We have an unmet need in the PEGylation, the urgent one. The two, I think so. The first one is the testing proteins. We are talking about the high throughput, okay? But there's no such a technology, I think so. So the first one is there's no versatile and effective method to screen proteins' pharmacological activities in vivo in a high throughput screening fashion. And the second unmet is that the conventional PEGylation technologies offer only limited half-life extension of peptide drugs in vivo till now. The first one is the A lot of new protein drugs has been discovered in these days and are subject to their feasibilities for their therapeutic value in various disease animal models. But due to their inherent short-lived and instabilities in the body, extremely high dosing regimens are needed and also fail to get expected data. Conventional PEGylations are time-consuming and not for in vivo high-throughput screening. The challenge is to develop a quick PEGylation to test protein drugs in vivo without compromising their biological activities with few milligrams for under 10 mice. We use the trail. Trail, they say, but the last blockbuster proteins, and it could be development for the cancers at autoimmunes and also liver fibrosis in the futures. But each biological half-life is extremely short, 5 minutes, okay, in rat and at the 30 minutes in human. So there has been no progress to clinical phases. We used well-established chemistry and reason that nickel acetic acid, NTA, chelated, has affinity to the histone-tagged protein. This is very common in the chromatographic purifications of protein. So we synthesized the pegylated nickel NTA and just incubated with histone-tagged proteins for this purpose. This method is just simple. Histone-tagged trails was just mixed with nickel NTA packs and incubated for 30 minutes and directly injected to the animal models. You can find out that the improved biological half-life and also pharmacodynamic in the animal models. This is cited in the NCI. And the second unmet need is very important. Extending peptide biological half-life. Okay, it's not common. Okay, many peptides are interestingly short-lived and unstable. Therefore, PEGylation is the method of choice for extending their biological half-lives. However, because peptides have relatively small molecular weight compared to proteins, peptides generally can only carry PEG size up to 20 kilodaltons. When a higher molecular weight PEG is applied to increase half-life, the conjugate PEG diminishes bioactivities of peptide drugs entirely. So, the question is, how can you utilize a high molecular weight PEGs on peptide drugs while retaining bioactivities? If you can maximize the half-life while maintaining activities in vivo, we can reduce doses and minimize also adverse effect induced by high doses. This is a simple the mechanism. Okay, the mechanism of action by comparing two pegylated peptides, each with different shape of PEGs, branched with a long space. As you know, extending force, the GLP-1 receptor agonist, was used on model peptide. As you see, the conventional branched pegylated peptide reduced binding affinity to each receptor, like this one. Okay, when a uniquely shaped PEG is used, it could retain bioactivities by increasing receptor binding. This figure shows the proposed mechanism of action of comparing to pegylated peptide, each with a different shaped PEG, branched, and the branched with a long space, but shift all of the two. Extending forward was used as model peptides. As you see, conventional branched pegylated peptide reduced binding affinity to its receptors. When a uniquely shaped PEG is used, it could retain bioactivities by increasing receptor binding. We think so. This slide clearly This means, okay, you know there's the new PEGylation platforms enabled, but we call that this is the first generations, okay? It looks the shape-controlled PEGylation. We compare the first generation PEGylations with another ones, okay? But the first PEGylation has a few advantages over conventional PEGylation, which means that the first, it's retained high molecular weight PEGs, such as 50 kilodaltons on peptide. This higher molecular weight offers longer half-life as expected with sustained release. Lower dose provide low risk of adverse effect. This cannot be done by site-specific PEGylation alone. We use the exendin-4s, you know, the GLP-1 receptor agonist. Exendin-4 is FDA approved. You know, everybody know about Bioetas, but it's a BID. And recently, Max Biodurance on the market, but I think that it's not not common in the clinical the field. I think so. But there's many approaches now on the market or on the progress. Okay. The current once-weekly GLP-1 agonist, the Biodurance, I mean that the microsphere versions, and that the Tangenium on albumin fusion approach and the duraglutide or FC fusion version are FDA approved, and also for lead compounds are under development as once weekly. However, there's no best-in-class compounds yet, mainly because of the high adverse effect and high production cost. Therefore, the market still, I think, need a better GLP-1 class compounds. We adopted 4G PEGylation technology to synthesize pegylated exendin-4 with high molecular weight PEGs, 50 kilodaltons. I think it's more than 10 times bigger than exendin-4 itself through a site-specific PEGylation. We call this Oledin. This slide shows another proof of concept evidence. PEGylation of exendin-4 with a high molecular weight linear PEGs significantly reduced biological activities in vitro as measured by intracellular cyclic AMP production. You know, in contrast, Oledin pegylated with a higher molecular weight PEGs but different shape, retained bioactivities comparable to conventional pegylated peptide. In addition, Oledin with 50 kilodalton PEGs retain GLP-1 binding affinity in GLP-1 receptor expressing cells. Almost, yes. When a subcutaneous injected in rat, as you see in the figure on the right, the peptide demonstrates significantly improved biological half-life, about 40-fold increase compared to the peptide alone. We should have studied PK in dog and monkeys. Thanks to the high molecular weight PEGs, Oledin showed the longest half-life in monkey, about 4 days, compared to GSK's albumin-infused GLP-1 Tangenium, LLC FC fusion GLP-1 compound. The PK parameter Oledin in rat, dog, and monkey are summarized in this. When Oledin was subcutaneously injected in type 2 diabetic DBD mouse, it immediately lowered the blood glucose and maintained glucose control for about 3 to 5 days, depending on the dose. We also test the pharmacodynamic on the Oledin after multiple subcutaneous injections twice weekly in DBD mice for a month. No problem was found. After 28 days treatment, Oledin significantly reduced hemoglobin A1C values compared to non-treated animals. Interestingly, Oledin treated mice showed normalized triglyceride values after a 4-week treatment. For translations, we have a GLP toxicology studies. No problem as expected. We tried to compare the anti-diabetic effect of orexin against Tenjium, albumin recombinant fusion protein GLP-1 agonist of GSK. As you can see in these figures, about 100-fold higher in vitro potency and longer half-life in monkey. And also, we expect more than 10-fold lower dose in human from preclinical PK and PD studies. Okay, we compared the ongoing half-life extension technologies with 4G PEGylations. There's many extend fusions, albumin and FC fusion method. Merit of 4G PEGylations could be found on the field of drug potency and dosage amount, resolving relatively high drug potency and also very low dosage amount against other technology. We are currently trying this 4G PEGylation technology with multiple representative biologics in my labs, and also recently we are collaborating with some biologic companies on the application of this technology to their interesting peptide and proteins candidate. I think the 6 months is good for the entire screening, I think so. Today, I talked about PEGylation and PEGylation technology progression, and also my lab's successful PEGylation extending 4 entry. There are a few new half-life extension technologies, however. Utilizing a PEG platform is the most practical way. From this point of view, we can say PEGylation is still a novel method of choice, particularly for translation R&D. And I think all the peptide and protein drugs must be evaluated their therapeutic values again by 4G PEGylation. I think so. Thank you for your kind attention.
Thank you very much. A few questions?
Okay.
Thank you very much for this nice presentation of I would say, up-to-date technologies for PEGylation. So I'm going to have maybe a little bit provocative question. Considering that, PEG is non-biodegradable and accumulates in tissues, may have chronic toxicological effects, do you think that compared to other technologies based on biodegradable polyamino acids or HSA, it's still a technology for half-life extension in the future?
The PEG is okay. We are always with polyethylene glycols, you know. But everybody talking about this the medications I received, okay. There's a problem on the safeties, but you know. But the 10 the drugs, okay, you know, the interferons and something like that, say on the market over 10 years, but you know. But every I talk about the toxicities, there's starting to appear the review of the toxicity. But I think the PEG is the last items to consider about the safeties. I think so. This is my question.
One more.
Thank you very much for a very thorough presentation. If you can provide us some explanation regarding the effect of the shape of the PEG change, linear branch star type, and what is the mechanism behind for observing this extended half-life of peptides, and what is the conformation of the change, of the PEGylated change with regard to the type and the shape?
Yes. I think that the receptor binding is the problem, okay? The linear is you know, PEG is very flexible, okay? Just a dance, okay? Around the molecule, okay? But each limit or the molecule size, okay? But the trimeric forms, for example, but with the same for example, the 50 kilodaltons, the size is very, very small. But we have used trimetic and binding to the receptor, okay? We attach that, there's on the market, the PEG, okay? The space size, okay? We controlled the space size, 1 kilo, 2 kilo, 3 kilo. We have data. But we have some, okay? No problem, I think so. That's the, in common. I'm surprised, okay, with the 50 kilodalton PEGs attached to the 4 kilodaltons, okay? It just still have bioactivities. I'm so surprised about that, okay. I think it is, okay, I'm happy to find it, okay.
Well, thank you very much. And now I'm following the exact protocol. I would like to invite Dr. Richard to hand over this certificate to Dr. Lee, and then pictures being taken of the two of us.
Okay. So, well, it's my great honor to give you the certificate of recognition from Omics International for this talk, amazing talk. And so I think we are going to have a picture.